Hello, good afternoon, everyone. My name is Avinash, and I'm a PhD student in Dr. Ori Rothstein's lab at the Keenan Research Center at St. Michael's Hospital. So today I'm going to be talking to you about a recent study that we have, this mouse study that we recently completed, really, and telling you about what the future mouse studies in this kind of mitochondrial transplantation field kind of holds in our laboratory. So originally, how we got really interested in mitochondria Ori had mentioned, which is that in hemorrhagic shock in our mouse model, we find that using remote ischemic conditioning, which is a therapeutic that we have well established in the laboratory, can increase mitophagy in animals. So then, actually, at a previous MITO2i conference, we came about this concept of mitochondrial transplantation from an invited keynote where you take mitochondria from a healthy skeletal muscle, and then you extract it, and then you give it to an organ that's experiencing ischemia, and this can increase the function of the organ, reduce injury to the organ. Okay, so here we really focused on liver ischemia reperfusion, and so I'll get into that. So first of all, what is the effect of IR on the mitochondria and mitochondrial quality? So IR is ischemia reperfusion injury. So here are some major aspects of what's going on in the mitochondria during this type of injury. There's large amounts of lipid peroxidation at the mitochondria. There's calcium release from the mitochondrial permeability transition pore. There's elaboration of reactive oxygen species. And this concept that has been brought about by all the panelists before, which is this elaboration of mitochondrial DNA into the plasma. And of course, we know that there's a depletion of ATP when you deprive the mitochondria of oxygen. So what is this mitochondrial transplantation process? Here I've shown you a figure from a paper where they discuss potential mechanisms of transplantation of mitochondria. These are not known and well-established mechanisms, but these are potential mechanisms. So you isolate them, and the current potential mechanism is that these exogenous mitochondria get engulfed by the cell into an endosome, and then they fuse with the mitochondrial network. And this can increase ATP levels, reduce oxidative stress, reduce apoptosis and inflammation. So in this manner, we decided to use a liver-specific model of ischemia reperfusion injury. So before we get to really what we did with the liver, we had to be able to get mitochondria from skeletal muscle. So we've been using this protocol from this paper from the Journal of Visual Science from Perbal et al. So how this works is that you take skeletal muscle from a mouse, and then you digest the skeletal muscle, you homogenize the skeletal muscle, and then you differentially filter it through different nylon sieves. And the last filter is about 5 microns. And after this, we spin down the mitochondria and we get a nice pellet. So we characterize this pellet to be protein pure. We blotted for ATPase, and we see that in the mitochondrial pellet, bottom right, there is quite a lot of ATPase and TOM20, which is another marker of the inner mitochondrial membrane. And there's a reduction in the amount of GAPDH, which is more of a cytosol-like predominant protein. And so we also wanted to know how good are our extractions. So at the top right, you see a graph with good R squared value, showing that, for example, from about, let's just say 1250 mg of mouse skeletal muscle, we can get over 1 billion mitochondria extracted. So this protocol has taken quite some time to get to this many extracted mitochondrial particles. And so how we count the mitochondria is we use something called a Coulter particle counter. And so that's helped us establish a way to reliably extract mitochondria and count them and dose them as if it was kind of like a drug that you can play with the dosage as much as you need to, and then inject it into animals to see what the animals are going to go through. So that's kind of the differential filtration process. So next, we wanted to see if these mitochondria that we extract had good amounts of ATP in them. And they did. So in the mitochondrial pellet graph on the left, you see that the pellet had a lot of ATP, and then the supernatant above this pellet had very reduced amounts of ATP. It's kind of a proof of the kind of filtration process. So we wanted to really know if what we were extracting is truly mitochondria that have intact cristae architecture, which is a hallmark of good mitochondria functioning. So on the right, you see electron micrographs. So the left parts where there's lots of white arrows are the active mitochondria. Each white arrow is pointing to a cristae intact mitochondria. And then we inactivated these mitochondria using liquid nitrogen by dunking the pellet into liquid nitrogen and taking it out 5 times. And so this totally disrupts the morphology of the mitochondria. So that's what you see on the right, is the inactive group. It's another control group to say that our differential filtration process can be destroyed by liquid nitrogen, really. And so here we are able to establish that there's good ATP and there's good cristae architecture. So what is the model that we're working with? So we work in surgical mouse models of liver injury. So previously, Ori's talked about how we've worked in hemorrhagic shock models in animals and mice. And so we wanted to have a liver-specific model now. So we essentially have a pre-ischemia period where we take the mitochondria from a different animal and then we extract them and we count them. And then we anesthetize an animal. And then we perform a midline laparotomy and we isolate the hepatic artery that's leading to the left lobe. And so then we clamp that artery for about an hour, and that induces liver ischemia. And you can very obviously see liver ischemia. And when you do this, the liver is way less perfused. And then we do a mitochondrial intrasplenic injection that delivers into the portal system, essentially, because there's a connection between the spleen and the portal system. And then we reperfuse the animal's liver for 1 to 4 hours, and we sacrifice the animal and we collect tissue and plasma to do a whole bunch of different kinds of analyses. So what have we shown so far? So this is what the piece of some of the exciting data that's been collected recently in our laboratory, which we are very happy about. And so the first part of this is plasma ALT. So plasma ALT is a liver injury marker. So we took plasma from the animals that had experienced liver ischemia, and we see from the left to the right on the left graph, the sham animals, which are just animals that have the surgery, but they don't get to have ischemia, have very low levels of plasma ALT, which means the surgical process is okay. And then there's liver ischemia, and you see a massive induction of plasma ALT. That's another control group. So we know that the model is inducing liver ischemia. And then the next group is the mitochondrial transplantation group. And we see almost a 4-ish fold reduction in the plasma ALT in the animals that have mitochondrial transplantation. So now it gets a little bit more complicated and a little bit also more interesting. So we took inactivated mitochondria previously shown by liquid nitrogen inactivation, and then we counted them and we injected the same number of them into the same types of animal groups. And so you see here that there is also a reduction in the plasma ALT levels, which is very interesting, that the inactivated mitochondrial particles that we inject is also inducing a form of protection of the liver. And we wanted to really confirm this further, so we inactivated them a different way. We inactivated them by heat treatment at 95 degrees. And then we injected these sorts of like protein aggregate mitochondrial particles. And then this also showed massive protection. But you see here that the mitochondrial transplant group, the ones that are active, are more protective than the ones that are inactive. So this is interesting data. Okay, all right. So what did we do next is that we took these things called polyethylene beads, and then we injected these. The reason to inject these types of beads is to really exclude a physical sort of interaction that's causing the protection. So then we injected these beads and we showed that another control group actually has increased amounts of plasma ALT, similarly to the liver ischemia group. And then there's of course the vehicle control, also having injury. So here we show that the transplantation process of mitochondria extracted from skeletal muscle is able to reduce plasma ALT and plasma AST, and this can protect the liver from injury in mice. Okay, so next, we really wanted to see what's happening in the liver tissue of these animals after we looked at the plasma of these animals. So that's why we did a whole bunch of histology of the same animals from the same study. So from the left to the right, it's the same groups that you saw in plasma, ALT and AST. First, there's the sham group, where we see there's very good sinusoidal patterning of the liver veins. And then you see the next group, which is the liver ischemia group. We see massive induction of like hepatopathology. We see the sinusoidal patterning like being destroyed. We see that there's a vacuolization in cytoplasm. So there's all these different markers of histopathology in their liver ischemia group. Okay, so, and then we, in the mitochondrial transplant group, we see there's very obvious massive protection of the liver histology. There is a few areas of necrosis still, like marked by light pink areas. So then this is quite a large kind of stark difference. And we've repeated this 6 or 7 times in different animals. And then we had a blinded observer who took all of these images and then ran them through the Suzuki score. And that's what you see quantified at the bottom here, where we see that when you do mitotransplant, there's a very stark normalization of the liver histopathology towards sham levels. And the 2 control groups, the inactivated mitochondria, are more injurious than they are in the active group. And the beads, again, are injurious. So here we show that the transplantation of mitochondria is able to normalize liver histopathology, and it's able to normalize Suzuki scores in a blinded fashion in a mouse model of liver ischemia. So in these same animals, we took their plasma, and then we did a few cytokine ELISAs. From the left to the right, that are the same groups as before. So firstly, we see a reduction in the amount of plasma IL-6 after you do mitochondrial transplantation. And then in the TNF-alpha group in the middle, we also see a reduction in the plasma TNF-alpha, which is another pro-inflammatory cytokine. And then we look at the plasma IL-10, which is an anti-inflammatory cytokine primarily. And we see that just by injection of something, increases plasma of IL-10 in these mice. So this is very interesting, right? So like it's weird, but it's also like you do any kind of intrasplenic injection and you see that there's an increase in plasma IL-10. But then on the pro-inflammatory side, we see obvious reduction in the pro-inflammatory cytokines. So from this set of cytokine ELISA data, we conclude that the transplantation of mitochondria is able to induce anti-inflammatory effects in mouse liver ischemia specifically. Okay, so at the beginning of the presentation, in the title, we had talked about efficacy, which we have shown using plasma ALT, AST, and the histology. And then we talked about mechanisms. So we did these groups where we injected the beads, where we injected inactivated mitochondrial particles, and we talked about translation. So in the translation aspect, we don't have data for this quite yet, but I just wanted to kind of show you what we're doing in terms of translation, which is that we're trying to have human livers sliced into very small pieces, which has been shown to be used as a tissue sort of model that can be grown in cell culture. And this is called ex vivo human PCLS. So this is very much in the works right now. And this is kind of a model that we're going to use to see if transplantation, or essentially co-incubation of mitochondrial particles, can increase or decrease function of these little pieces of human liver tissue. So it's like a human aspect to the project. So finally, I would like to thank a whole bunch of people who have been central to this work. My supervisor, Dr. Oya Rothstein, my committee, Dr. Kappas, Andriatsa, and Dr. Jeschke. And thank you to the entire laboratory group. And thanks to MITO2i for this opportunity to present some of this recent data that we're very excited about.
Thank you very much, Avinash and Erica. It was a great presentation, and congratulations to both of you for being the awardees of this scholarship year for MITO2i.